Thank you to Gregor Bucher for joining us today for our AgBio Data webinar. I get to introduce Gregor. I know him actually. We are part of the I5K Insect 5000 Genomes organization where we work together on common standards and coordinating genome sequencing projects, and where we're part of a working group there. But in his regular day job, he's a professor at the University of Göttingen in Germany, where he works primarily on Tribolium castaneum, right, Gregor? So the red flower beetle, and specifically on the genetic basis of development and evolution of insect head and brain, and also new modes of pest control using RNAi. So there is a bit of a ag focus there too in your work, Gregor, in addition to traditional evo devo type work that his lab does. And his lab is also working on developing new transgenic tools for emerging model organisms such as Tribolium. And where he's going to segue in today for his talk within AgBio Data is that he initiated and led the first genome-wide RNAi screen in an insect model outside of Drosophila melanogaster. So it's kind of a big deal for us working on and wanting to take advantage of functional genomics insights in insect models that are not from just one organism. So it was a great resource, and he established the iBeetle-Base database as a result of collecting all that data for that RNAi screen. And that is going to be the focus of his talk today. So for that, I will turn it over to you, Gregor, and you can tell us about your database and the lessons learned there. Quick question before you start, though. Are you open to having us? We're a pretty small group today, it seems. So can we keep this more open where if people have questions, we can just go ahead and interrupt you, or would you prefer to just do your thing and then we'll ask you later?
No, sure. I'm happy if you interrupt me. I will not be able to follow the chat, so just unmute and shoot your question at me.
Gotcha. Okay, thanks. We'll go right
Yeah,
ahead,
thanks for the
Gregor.
nice introduction. And I think Monica put it very correctly that actually I'm not really in the agricultural field and also not in the database field. I kind of slipped into that, and this is kind of what I will also show in my talk. Okay, so does this work? Yeah. So what I would like to say or tell you today is about insect functional genomics in a more general term and why we're using the Tribolium model system. Then that we performed this large-scale RNAi screen to find genome-wide information on phenotypes. And I will also sneak in some information about the best target genes for RNAi-mediated pest control, which is kind of related to that project and might be of interest to some of you. And then I come to the core, which is iBeetle-Base, which we had to establish in order to make this genome-wide data available. What challenges we found, and then also what functionality it has, and then also the vision what we're trying to expand from here. So if you want to understand the gene function, it's not enough to just sequence the genome and see all the components, all the genes of a genome. You really have to find out what they do. And actually, most of the components, most of the genes of an insect, if you mutate them, don't have a phenotype, right? So at least in the lab, they just survive, and you don't know what they're really doing. So no, sorry, 40%. If you mutate 40% of the genes, you don't see anything, which also means that 60% they will suffer or even die. So if you knock down a component, then you find out, okay, if this gene is mutated, then at high velocities, the organism is kind of derailing. And then you know, okay, this is an interesting gene. It's required for remaining on the street. And this is kind of the basic understanding of gene function. You have to knock down a gene, you have to reduce the gene function in order to find out what it actually does. And this is now an example from our lab. This is an insect larva with legs, appendages, and antennae. And if you knock down the gene distalis, these things get shorter and even almost reduced. And based on such experiments, knocking down one gene, seeing the phenotype, you would then say, okay, this gene is responsible for the outgrowth of, or is required for the outgrowth of the appendages. And let's call it distalis because in the mutation, the distal parts are missing. And this is the basic concept about gene function research. And since the advent of RNAi, this can be done not only in the big model systems like flies and mice and so on, but also in many other insects. RNAi essentially, if double-stranded RNA gets into the cell knows this is something evil. The cell knows double-stranded DNA, that's okay. Single-stranded RNA, it's also okay, but double-stranded RNA is bad because it usually comes from viruses or from transposons. And the cell cuts it into pieces, puts it into the RISC complex, which then will lead to a destruction complex, which is going around the cytoplasm trying to find um, messenger RNAs that are homologous and then cutting them, destroying them. And we're using that, this defense mechanism of insects we're using for studying gene function. In our beetle, we are lucky, so the cells take up double-stranded RNA from the environment. So what we, for instance, do, we inject double-stranded RNA into the hemolymph of insect, of larvae. The cells will take up the double-stranded RNA, will knock down the gene in the cell, and then we can see what happens during metamorphosis if a certain gene is knocked out. Or even further, if we can inject female pupae with double-stranded RNA, mate them, let them hatch, and then we can look at the offspring because the RNAi effect is also transmitted to the offspring. So in former times, it was like that almost everything that you knew about gene function come from this one insect here, Drosophila melanogaster, because these classic screens could be done only in this species. But now that RNAi is available, you can also study gene function in all kinds of other organisms. And what has been done mainly predominantly in the last 20 years, let's say, was to see what genes are known to be important in flies, and now let's test them in other organisms, which is what we call the candidate gene approach. Of course, this is a bit limited because you can only find conserved functions because you come with a assumption. You come with the assumption, okay, this gene was important for for wing development in flies, so maybe it's also in beetles, but you will not find genes which are important for beetle elytra development, but not in fly wing development. And we are using the red flower beetle, Tribolium castaneum, for our work. Biologically, it's interesting for us because it has a more ancestral, more insect-typical type of embryogenesis. This is why we want to study this organism as compared to flies. It also has an ancestral type of metamorphosis, which is also kind of more interesting to see how holometabolous insects have evolved. And of course, beetles are the most species-rich taxon on Earth and also have many pest species. So it's a very good model for evolutionary diversification and success, and also for pests. There are also a number of practical reasons. It has a very short life cycle, 3 weeks, which is really good if you want to do genetics or transgenic tools. It's easy to keep. We just keep them on flower. They don't even need water or fruit or something. And we have a number of really quite advanced tools there. So we can knock down gene function with RNAi. We can do genome editing, kind of inserting GFP, or we can insert transgenes. So with respect to studying gene function, Tribolium castaneum is pretty much the, let's say, second best insect model system that we have currently. Okay, and I told you that for a long time people have kind of used the genes known from flies and tested them in beetles. And at some point we found, okay, we now have tested all of them, but we know that we are still missing things. And then we wanted to find the remaining genes that are important for development, for embryonic development, for instance, in beetles. And in order to find those genes, we initiated this large-scale screen iBeetle. So actually we did two screens. As I showed you, we injected female pupae to look at the phenotypes in the offspring. This is a test for for gene function in embryogenesis. And we injected larvae in order to see what happens during metamorphosis. So this was all focused on the development of insects. And we used transgenic lines that had muscles marked with GFP in order to also check for defects in muscles. This is a brief overview on the procedure. So we injected 10 female pupae of that transgenic line. And at day 3, we had another look at them because they should have hatched by then and wanted to see, do they have, are the adults normal or are they dying? Are they melanized or something? After some days, they started to lay eggs, and then we made a cuticle prep to see defects in the embryos that are laid by these injected mothers, because as I told you before, the RNAi effect is transmitted from the mother to the offspring. Then we prepared a second egg lay and looked at defects in the muscles. And if in this egg lay the number of eggs was highly reduced, then we also dissected the ovaries to see if there's some defect in oogenesis. This was a very complex procedure, so we were able to screen 21 genes per week per screener over 1 year or so. And I should say that the injection is actually not the limiting thing. The limiting thing is really the analysis, this really complex analysis that we have done. I want to show you one example where we found new genes that were kind of unexpected. So in Drosophila, they have observed years ago this wing blister phenotype. Essentially, the wings are two epithelia. They are attached to each other by integrin. And if this integrin attachment doesn't work, it can kind of detach and form these bubbles that you see here. These are liquid-filled bubbles. This is called the wing blister phenotype. And these are genes which are important for muscle development and so on. And this has been studied quite a lot. And we also found these kind of wing blister phenotypes in our beetles. So just to see if we are able to find new genes for this, let's say, well-studied process, we looked at wing blister phenotypes from the iBeetle screen. We found 75 of them. We chose a selection to confirm them and could confirm with non-overlapping fragments. And then we asked, are these genes that we found here that have not been reported from flies, are they really also maybe also acting in flies? And indeed, what we find that 19 of these genes we tested, and several of them really also did their job in flies. 5 did not, but all the others did. And this is the most important thing. 6 of these genes had no functional annotation whatsoever. So this was really completely unexpected genes in that process that Drosophilists might say was well studied.
Gregor, I have a question, if I can
Yeah,
interrupt,
sure.
since we said I can interrupt. So that's super cool. Did Flybase, were they able to curate those genes then? So sort of a round trip process where you
Mhm.
were initially getting into functional genomics and tribolium, but then the fly literature was able to use what you had initially wanted to study in tribolium to enrich the Flybase data set.
Mhm. So if they used this data, this is published, this data here, so they might have entered that, but I'm not sure. We haven't really kind of asked them to do that. What we did is for all the, let's say, 200 genes that we really studied carefully with non-overlapping fragments, all the controls, we determined GO terms for tribolium and submitted this to the GO consortium. So the information should be now available. If you're searching for GO terms, most of them come from flies and mice and whatever, but some of them might also come from tribolium then. Flybase, they were willing to link their genes to our gene information page, but they did not kind of insert our functional data from beetles for flies. I mean, which makes sense, I would say.
Although that was actually then for fly, the secondary analysis that you did to follow up in this
Exactly.
case,
This we
right?
did in
Yeah.
fly, but this was just seeing, do we see the phenotype or not? But this could be entered in fly as I'm not sure
Mhm.
if they were realized that this data is out there.
Yeah. Okay, thanks.
We studied a number of developmental processes and tried to see, okay, how many of the genes were identified in flies and beetles, and this was around 50%, let's say, over these 3 or 4 developmental processes. Then there were 10% of genes that there were clear phenotypes in flies, and we did not see any phenotype in beetles. So this really seems to be biological difference. And there were 40% of the genes where we saw a very clear and reproduced phenotype in beetles, but not in flies. And I think this is a mixture of biological divergence on one hand, and on the other hand, a technical one, because we really have a genome-wide data set. While in flies, they do, for instance, these genetic screens, find 25 mutants in a certain process, but publish only 5 of them. And the others are actually doing something, but it's not published, so it's not in the database, right? So maybe we are a bit more comprehensive now with our database because it's really gene by gene, more or less. Okay. And actually, this was all aimed to find new developmental gene functions, but we also found genes where the beetles just died, right? For developmental biologists, this is a disaster because you cannot see anything. But of course, for people interested in fighting pests, this is very interesting. So we kind of deviated part of the lab into this direction. And we did together with Bayer, a large-scale screen to find the best RNAi target genes for pest control. Okay, so the idea of RNAi in pest control is to spray double-stranded RNA on plants. The pest will eat it and will die because this will be a double-stranded RNA targeting an essential gene from this beetle. But bees would not die because this would target only a beetle gene but not a bee gene. So in principle, this mode of pest control is species specific, and the substance is completely biodegradable, so it's eco-friendly. The question is, what do we use as target genes? Or what are the best genes to target if you want to kill an insect? And if you have around 16,000 genes of an insect, as I told you before, 40% lead to lethality, meaning it's not so difficult to find a gene that kills an insect, right? Many genes will do that. The question is how well it does that. And these are two examples. This is VATPase, which is one of the quite known genes to kill insects by RNAi-mediated pest control. And what you see that this is the number of or the portion of animals that are living. So it starts close to 100%, and then over time they all die. At the highest concentration, they die quickly. At the lower concentrations, they die more slowly. So this is what this lethal gene known from other insects does in beetles. But we found, for instance, a gene which does the same, but it does it much more quickly and much more efficiently, and also at the lowest concentrations. So the challenge is not to find a lethal gene. The challenge is really to find the best lethal genes for application. And if you think about spraying double-stranded RNA on acres, every 10% increase of efficiency will, of course, reduce the cost. So it's really important. So people have started to think about and read literature to find genes that are maybe good targets. And the expectation, of course, is you have a transcript, you do RNAi, the transcript goes down with some lag. Also, the protein will go down and the insect will die. That's the expectation. However, there are some things that interfere with that expectation. For instance, if you have a very stable protein, despite the fact that RNAi was efficient, it will take a long time until they die. Or sometimes we have paralogs, which may be able to compensate for the knockdown of one gene. So the paralog is kind of upregulated and will at least partially rescue the effect of the one. And sometimes we also have regulatory gene networks where the downregulation by RNAi leads to an upregulation, such that in some you don't reach this downregulation, but actually the downregulation is not so high. And this is all pretty much unpredictable. So we don't have the data to really predict which gene would behave like this and which one would behave like this. So, and if you cannot do it by knowledge, by reading literature, what you can do is to do an unbiased large-scale or genome-wide screen. And of course, doing a genome-wide RNAi screen is not good with pests that you can work with only, let's say, 2 months or a year or so. And this is why we did this genome-wide effort in our red flower beetle, which is really a cool model system and easy to keep with lots of offspring and short generation time. So how did we do that? We collected L5, L6 larvae put them on ice to anesthetize them, put them into these little slits here to inject them, and then we injected 10 larvae per gene, put them back into flour. This was all color coded in order not to get confused. We put them into incubator, and after 1 week, we just counted the number of dead animals, put that into our scoring sheet, and then in an Excel table, and then we could kind of make a list of lethalities of these thousands of genes. And here the throughput was much higher. As you remember, in the iBeetle screen, we had 21 genes per week per screener. But this is the most easy screen that you can imagine. You just need to inject and to count dead animals. It can be more easy, and we managed to do 156 genes per week per screener. And the screener who did that for 2 years, and it didn't get crazy, right? So it's really something that you could speed up, let's say, by some percent for a few weeks, but maybe not for years. So in the end, we managed to test this for 15,000 genes, which is close to 95% of the genome. And then we said, okay, from the entire set, we will use all genes that after 7 days led to at least 90% lethality. And these were kind of 1,000 genes, kind of 6% of all the genes. Then we did some more experiments, which I will not go into, to extract from these 900 genes the best one of those. And we came up with a number of about 200 most affected target genes in our beetle. And then we looked if we can transfer that to a real pest by feeding. So this was, I have to say, this is all by injection. And we wanted to transfer that to feeding. We tested not all of them, but just 66, and 50% of them also were highly lethal in another beetle by feeding. So these then we called our 34 superior target genes. So they were better. And then we used some of them to test in the next, in another pest. We tested 12, and most of them really did it. So in the end, we can say, okay, we found many genes that are effective by injection in tribolium. We have identified 34 which work by injection, but also by oral delivery, and also something which worked in 3 species. So this is kind of what we would say. We have high confidence superior target genes. We have a larger set, and this is something that we now contribute to the community that they can test this for their pest. This is one example. The cabbage stem flea beetle starts being a really severe pest in Germany, at least. And we tested a number of these genes that we knew here. And if we now look at the subset of 34 superior target genes, you see that most of them are the most lethal genes. There are also some, for instance, this one or this one, which are just as good, but are not part of that. But most of the really strong lethal genes are part of that subset. And if you look at what genes are actually the most lethal genes, the best genes for RNAi-mediated pest control, it's really all highly conserved cell biology genes like proteasome, like protein degradation or translation protein production. It's all kind of very basic cellular processes.
Gregor, I
So
have a
this,
question.
yeah,
Sorry,
sure.
I can interrupt again. So, that's a good point, but wouldn't companies also want to understand species-specific lethality genes? Because, you mentioned that Coleoptera is a hugely diverse order, and so there's probably some species there that are of conservation concern. And so there might be cases where, you'd want to have knockdown or lethality in the pest species, but you wouldn't want off-target effects within even that same genus. Did you also look into that aspect of it as well?
Yeah, that's a very good point, very important one. And luckily, most proteins are not conserved throughout the entire protein sequence. So you will always find in the messenger RNA a stretch which is not so conserved that you wouldn't be able to find a region to target without targeting others. Um, we went a bit into that, and I think the bottom line is it's not difficult to find a region that targets your pest species and not the most important beneficials that are around. I wouldn't say you can do it in a way that it doesn't target anything else, because if you sequence enough species, you would have no problem. But it's actually not much of a problem, I would say. So you have to do it bioinformatically. And actually, DOA has programmed a web page where you can kind of do that. And then it will optimize for least number of targets in other species. So I would say if you're developing a product, you will be able to find a sequence that targets your species very well. The beneficial, let's say 5 or 10 animals which are around in that region, not at all, and all the others almost not. Okay, this is maybe the first take-home message. The best targets for RNAi-mediated pest control are really basic cell biology genes. There is no such thing as the best target gene for all. So this is really species specific and has to be tested empirically. And the suggestion based on our work would be, depending on how well your model organism, your pest works, test at least these 11, or if it's more easy, test the 34 that we have kind of pre-screened. And it's quite likely that you will find a very good target gene for your pest. Okay, but I'm here for the iBeetle-Base. So this is now what I'm spending the rest of the talk. The first was we wanted to annotate phenotypic data, like morphological phenotypic data. And this is not so easy because there are many different ways to describe such a phenotype. And we tried to follow this so-called EQM system, which means you first name an entity, like a wing, the quality, like the size, it could also be the color, right? And then the modifier, in what direction this quality is changed. For instance, here you have a wild type larva with a wing, and here you see the wing is much shorter, so you could say wing size decreased. This would be entity quality modifier. The same here, leg color lighter, right? So you see it should be brown as the similar to the wing, but it is not. It got lighter, so something happened here. So this is a theory, and we try to do that, but we also realized that you can also annotate it like wings shortened, right? This is just the same thing. So even if you have a controlled vocabulary in your database, still there are different ways to correctly describe the same morphological phenotype. And even as we tried a lot to standardize the way we annotate, I have to admit there's a lot of variability that, let's say, a human being would notice that wing size decreased is the same as wing shortened, but let's say a simple search function would probably not. So this is the challenge. There are different ways of describing morphological phenotypes, and it was really difficult to standardize the manual work of 15 different persons who had a different way of describing things. So I would say we tried our best to standardize it, but it's not well standardized. So if you read it, you would recognize that it's relevant, but the search function wouldn't work. So what we then recommend to our users that they should just search for the entity, for instance, wing, and then have a look at the pictures to select those phenotypes that they are interested in. The other challenge that we encountered is that on one hand, we would like our screeners to be as specific in their annotation as possible and to really name the substructure that is affected. For instance, here, this is annotated as larva trochanter size decreased, okay, because the trochanter was much shortened. Or here, in the adult, tarsus not present. But now if I'm looking for leg phenotypes, I would enter in my search function leg. and I would not find trochanter and I wouldn't find tarsus. So if I now would like to find all the phenotypes that are affecting a leg, I would have to search for coxa, for trochanter, for femur, tibia, tarsus, and pretarsus, and all possible structures that are there. Yeah, Leonore, you have a question?
Sorry, yeah. Whoops, I just turned the video off again. So this to me is like, is there not like a beetle anatomy ontology? So, oh, okay, because that would sort
Okay.
of solve your problem, right? That you have like the femur and the coxa are all part of the leg, and so you could make use of that
Mhm.
ontology structure.
Yeah,
Oh,
we should have
okay.
talked to you before we started the database because this was exactly what we had to do. So we uh, had to make an ontology where, let's say, for instance, a trochanter, we know that the larval trochanter and also the adult trochanter both are trochanter as a concept. And the trochanter is a podomer which is part of the leg. And if I search for appendage, the computer would be able to kind of see, okay, everything which is annotated with trochanter, coxa, femur is a leg or an appendage.
Is
We
there any representation like this in like Uberon? You know, in the sort of meta um, anatomy ontologies, or no?
um, they tried to
You
put it somewhere. I'm not the ontology expert. But I think I heard Oberon somewhere. Yeah. But I'm not so at least they try to be part of that universe. We tried to use fly ontology because this is of course very extensive, but we soon realized this is not possible because, for instance, flies don't have embryonic legs, uh, and beetles don't have halteres, but flies don't have elytra, so we were just not able to copy paste it. We also tried to find ways to coordinate with the arthropod ontology, but this is really it gets really difficult if you really want to find terms that are really overarching, important. It's really, and I don't know if they still are working on that. So the idea was then, okay, let's just do one ontology for our purpose because we need it now. Let's then map this one to the arthropod ontology, which would then also be mapped to flies in order to make that. But I have to say, I haven't followed the arthropod ontology further, and I cannot say if this is still ongoing, and we haven't also not mapped them, unfortunately. That's also one of the challenges, right? There's many things that you should do, but then you need money for that, and yeah, you know. Okay, so these were the difficulties that we encountered, and now I want to present you the database. So so really what the special thing is that we have the phenotype annotations here. So this is a search function, which is really very simple. You can put in IDs like the tribolium gene numbers or fly-based numbers or fly gene names, but you can also look for phenotypes. What I would kind of recommend to just look for the entity, abdomen or wing, and then find phenotypes. Then we can also go to a genome browser where we would see for all the genes that are there. We have the genome browser with all the RNA-seq tracks and so on. We also have a functionality where we can transform Drosophila gene numbers into tribolium numbers or iBeetle numbers into Drosophila GC numbers and so on. So you can kind of choose the transformation that you would like to do, and then this conversion pipeline will kind of do that for you. Then, of course, we also offer download of other resources like links to the tribolium community, download the genome assembly, and so on. Okay, this is the search for genes. And if you search for a gene, it will suggest what there is in the database. Then you click on this, and then you have first the overview. What is the gene identifier? You can have this link to go to the genome browser and see the environment. You can download mRNA sequence, coding sequence, and so on. And we also have outlinks to Beetle-Atlas, which is a large-scale transcriptomic resource, or to AutoDB, which also kind of provides orthologs. Then we have a little screenshot of the genome browser, and also we provide information on the closest fly orthologs. Then more at the bottom of the page, we also show the phenotypes that we obtained in the screen, and we show the pictures and also the annotations that the screeners have done. For instance, labium not present, 100% penetrance, right? So somebody looking for these phenotypes will be able to judge based on these information how important that would be. Okay, I already told you, we also have the outlink to Beetle-Atlas, which kind of documents the expression in different tissues. And we also can search for phenotypes in this search field. For instance, if you type in leg, it will say, okay, leg arista wing complex and so on. And then you will have 151 phenotypes, for instance, and then you can choose here, I want to have genes that elicited the phenotype with high penetrance, reducing the number, or with low penetrance, kind of increasing the number. And then you will see the gene, the annotation, and also the pictures that are associated to this phenotype. And for instance, you can add to this leg also the stage, and then you will not see pupil or adult legs, but only the larval legs. Now comes something which we find might help in kind of curating databases with small communities. So we have a community edition. So the community is allowed to upload data. One type of data is publications. So when you go to a gene information page, there's also this button, add a publication. And then you just put in this PMID, and then this publication will be linked to this gene. We also can submit geoterms. So you would kind of propose a geoterm. So these are the geoterms that are from flies, but also we have functional data, so we can propose a geoterm based on tribolium data. And if we do that, then there are a number of fields that you have to put in, the evidence code, the publication where it's in, and also the respective quotation in order to kind of show people quickly what we have found. Finally, we also allow the community to add RNAi phenotypes, such that we have not only the phenotypes that we found in our screen, but also the individual phenotypes can be uploaded. With all kinds of fields that are important to reproduce that experiment. Okay, so now let's come to the vision, what we kind of think the database might do in the future for the community of people working on gene function research. Because this is really a problem. We have many ways of showing genome data and interlinking that I5K, like all these initiatives. NCBI is really great. But these databases only show, let's say, the genome view, right? Transcripts, proteins, genomes. And there are only a few databases that really refer to gene function in the sense of what happens if you knock down the gene, what happens to the animal. And of course, FlyBase is the most important one. Now we have Beetle-Base, much smaller, but still valuable. But for all the other functional model organisms that are out there, let's say 1 or 2 dozen or so, there are no databases available. Mainly because it's really a lot of effort to program a database, to manage the hosting, to update it, to filling it with data, and to curate it. This is really where you need many persons, and these small communities usually they consist of 1 or 2 or 5 persons. So it's completely unrealistic that they do this. So the idea is now we have a database that does that, and we have a database where the community can enter data. So we can reduce this problem here of programming, hosting, updating by just cloning iBeetle-Base for Oncopetus, for Spider, and for others, and really making sure that the code is identical and to host it at the same location, meaning that if we host one, we can host five, right? So this is then for the other communities, there's no cost and no effort here. In normal databases, you have dedicated curators who enter data and the community is just kind of reading out the data usually. Our community edition would allow that the community itself is adding data and reading it out, because we will in most cases not be able to finance dedicated curators for that purpose. So this would also solve the problem of filling in data and data curation. I don't know if this is realistic or not. We're just in the course of applying for money to do that for, let's say, 5 other species. And we'll try to see if we get the money and then how well this works. So the take-home message 2 is we have many standard databases for omics, but not so much for phenotypic databases, really showing the function of a gene. The vision is that we can overcome this problem of the small communities by database cloning and by community addition. The problem is, how active are the communities? This is a bit questionable, let's say. And the idea, the vision is that we have a phenotypic database cluster for these, let's say, 5 or 10 or 12 model systems where a lot of RNAi work is being done. I really would like to thank 2 persons which were really key for the iBeetle-Base. Jürgen is really Mr. iBeetle-Base. Without him, this would not have worked out. He's also the Mr. Ontology. So he really, and he kind of like Leonore, he immediately saw we need an ontology and convinced me. And Gang, actually, he's the one who did the relaunch of iBeetle-Base, and I hope to hire him also for the next phase. And with that, I would also like to thank you for your attention, and I'm very happy to take questions.
All right. Thank you, Gregor. Give them a virtual round of applause here. So, questions. There are a couple on the chat that I'll start with, and then we can open up. So Tanya has the first one, Tanya from TARE. So could you talk about the uptake of Go annotation submission through your form? So that sort of community-based interaction, how's that going for you? So both Go and the RNA phenotype additions from the community.
Not exactly sure if I understood correctly. So what people would do, we would
well, how's it going? Like, how much uptake do you have?
What do you mean with uptake?
Has it
that
been
people
used?
do it?
Yeah, do people
Ah,
do
Yeah,
okay.
it?
I think that's what she means.
Yeah, okay. I understand this is a key problem. We have the opportunity, but many people are willing, but then they don't find the time. So I have sent around in the iBeetle-Base, and so far we have about, I think, 30 or 40 entries, which is quite good, I would say. I think on the long run, one would have to kind of tell people, okay, let's do a joint paper. Everyone who enters more than 5 entries will be co-author. This might be something that then helps people doing it. And I should also say I had some money left from the iBeetle project, and I also then paid a person to do that. Yeah. But that's probably the main uncertainty with this community contribution. How active are the communities actually in doing this?
That seems to be a common problem, not just limited to beetles, but everywhere, right,
Yeah.
Tanya?
Yeah,
Everyone
and
wants
as
the
Lenore,
data,
as
but they don't
Lenore
want to spend
followed
time
up,
on it.
yeah, and as
Okay.
Lenore followed up, we have this community submission too, but there's a real variability in the quality of the submission. And so sometimes I think it feels like we're spending more effort reviewing than if we had just done it ourselves.
I see.
Is that
I mean,
an experience you've had as well, or maybe your community is like more gung-ho and more?
mm, I have to say I haven't reviewed carefully enough. I mean, the most things were contributing papers, and this is actually kind of simple. This is standardized, right? Um, I haven't reviewed the other contributions, and I'm not aware of many people contributing RNAi phenotypes because this is probably the most complex task where most things can go wrong.
Mhm.
And we also encountered one issue. So first we thought we have to force people to enter some things to be really complete, but then people kind of were almost done and then they didn't have the data for this enforced field and then they would stop. So then we kind of said, okay, let's enforce less in order to get more data, even if the quality may be less. So this is a difficult thing. But I also should say we don't have too much experience so far.
Eleanor?
Okay, I'll just sort of jump on that and just say, I mean, I think Tanya and I would probably be very happy to share sort of what our experience has been. And also, I'm going to be going to this ISB meeting coming up this weekend, and there's actually a whole session on community curation. So there might be relevant information about that. And it also occurs to me that a micro publication might be the thing that you need for some of these annotations. So micro publications are these like single figure papers. So, I mean, it's an actual publishable unit like that. You know, if they're contributing functional annotations to your database and they get a paper with a DOI and all of that, like that's a citable unit that has a little bit more currency. And I can happily put you in touch with the micropub people there. Tanya's putting in the chat.
Yeah, that's a great idea.
Some of the members of AgBioData, I mean, they are an affiliated organization to AgBioData, and some of the people, like we curate Arabidopsis papers for them.
Okay,
So
that's
yeah.
cool. Yeah. Yeah, good idea. I was thinking about a joint community paper, but of course, if you have micro publications, everyone gets the credit, then it's kind of might help. Yeah.
Yeah, Flybase is one of their partners too.
Cool. Thanks.
Another group I've heard reference when we're in the conversation around successful community annotation efforts is PalmBase. So it's I think that's what they're called. So it's I forget what yeast species I should know, maybe Candida albicans or something like that. But that's a model of a successful group that managed to galvanize the community, have web tools that really allowed them to streamline community annotations and sort of the educational platform to tell people how to use the forms in a way that the quality of the annotation was all right. I think it's Canto. Okay, that's the platform. Yeah, that might be something to look into. But yeah, if you figure out the community uptake problem, like let us all know. So, we're on that. Okay, so Lenore had a question for each of the communities. Would they also need so thinking about that clonable database aspect where you expand beyond Tribulum castaneum and move it to a different beetle or other insect species? To what extent would you need bespoke anatomy ontologies? Would the different organism need individual development of a new ontology or expanded one?
I think we would need that in order to make phenotypes searchable. But this is, of course, also quite an effort, let's say. Yeah, that's an unsolved issue. I mean, if we don't implement it, then we would have just a reduced searchability, which may be okay for the other organisms because we don't have much data there, right? So it might be enough to just search for, yeah, that's something that we have to think about when we clone it, right? But this is really unsolved. We cannot just take over because elytra is something that only beetles have, right? So we would need this arthropod ontology to really do that. That's a project on itself, more or less, right?
It might make sense to evaluate incorporating the existing ontology that you develop into Uberon, so that then you would have the kind of the overarching infrastructure, and many of the structures in the new beetle or new beetles that you have would already be there. And then you'd only have to add, I don't know, 10 more structures instead of having to create
Mm.
the whole next beetle ontology for the next
Mhm.
project. It's not zero work to incorporate it into Uberon, but I think if you're going to clone and need more and more parts, that may pay off. And then you're in
Mhm.
the world of that larger shared ontology.
Mhm. Which also should be the way to go, right? That we go away from this
Yeah, and
isolated
then there's a
efforts.
larger community that maintains it, right? So you're not on
Mhm.
your own
Mhm.
anymore.
Yeah, yeah. The question
You
also
know,
is, I mean, how much effort do you spend in an ontology where only three phenotypes will be, right? So if you have hundreds or thousands of phenotypes, it pays. If you have 1 dozen of phenotypes, how much person months would you spend on something that is not used, right? So that's also the question. Yeah.
yeah. I also think that they might have experience in mapping external ontologies onto the main one. So there are other communities that have developed their own um, anatomy ontologies that have been incorporated into Uberon. And
Hmm.
so
Hmm.
there may be a process in place to take most of the terms over,
Hmm.
so
Hmm.
that you don't have to start from scratch. So it might be worth investigating.
Hmm.
I do think it is in general a big problem slash opportunity slash big grant proposal to, tackle the question of we have all these new, reference quality genome sequences. We have these tools that allow in insects and probably plants too. This shouldn't be unique to insects for non-model species to be able to identify unique, interesting phenotypes just piecemeal and across thousands of different species. So like moving away from we're going to characterize everything in a model approach to let's have bits and pieces throughout the phylogeny that might be useful to inform phenotypes and related species or even back to the models. And so what's the infrastructure to actually facilitate that? Um, and I don't know that has anybody answered that question or is developing something? Because I think that that's something that's needed and that we've thought about too, but haven't had the bandwidth or capacity to really figure that out. Because it's going to happen. And then how do we actually make that data more useful across the entire all of arthropoda or within plants? It's not going to be unique to insects. Um, it's sort of a general big question, big picture question. And if you're writing your DFG grant proposal, maybe you should put that in. But, as in plants, like for the plant group here, has that question been addressed? Is it there also? Are people working on infrastructure to handle this or?
I just tossed something in about like Zach Littman has published on like a sort of whole pan speciesome. So like actually like pan Solanaceae genomes. I haven't really dug into that, but I suspect that's sort of the directionality. So you have all of the different genomes from representatives of all of the different um, species within that group. I'm not sure where that's kind of going from there, but
I
I
mean,
was just
I
thinking
followed
that's
a
something
bit the
close.
efforts from the arthropod ontology, and it was really they really met every month and were discussing and everything. But it's really slow because if you really go into detail, it's really getting very difficult. And you need experts in morphology of all that. So it's really not it sounds more easy than it is actually. I mean, I think somebody said that the high level annotation, I mean, this would be possible, I guess. So this is something that we could probably do, right? And the other
Yeah.
problem actually I see is that, I mean, we're interested in morphology of larvae and pupae and adults, but others might be interested in physiology or in brain structures or in gut, whatever. And if I want to provide an ontology for everything, I would spend the next 20 years on that, but I don't know what they need, right? And so it's yeah, really difficult.
or find a system where it's just piecemeal. I'm just interested in this particular part of a larval leg in this species, and how can I just pop that ontology term into a larger structure and then go off, do my business, and move on from to describe the entire, phenotype or ontology to describe the entire phenotype of a particular species. Um, Well, we're at the top of the hour. Will be one more question. Tanya had a question on the software stack, right? I think I saw that somewhere. What's the tech stack
She
for
did,
these?
but she had to go.
Oh, yeah.
I have to confess that I cannot say anything on the code, so I would have to ask Jürgen. He knows. He would be able to
Yeah. That's
answer
a
these questions.
sustainability question, though, too, is, how do all these databases now with variable funding in different areas of the world be able to sustain ourselves, and that sort of clonable solution might be interesting sustainability wise. So if you're interested in like talking to the sustainability working group about that as, something to, especially with the European funding base.
So we have discussed this. So we have an IT provider who was actually hosting it, and they promised to maintain it in the sense that if a new component comes or the system is updated, that they will make sure that it will run after system uptake. I think we're really lucky to have them here. And they also wanted to see what components we're using in order to see if they would be able to or willing to maintain it. Then there was the idea, first we had this Java server interfaces thing, but then people said this big thing does things that are not meant to happen there. Then we kind of removed that and had many microservices. But then people said, if you have too many microservices, we will have a lot of work to maintain each of them. So yeah. And then you don't know, do you take a system that works, which is maintained by one person who might stop maintaining it, or do you use something else which may be less fitting, but backed by a community where you think the community might still exist in 5 years? That's really
Yes,
difficult.
yes. All the problems. So we can continue to know good conversations in Ag Biodata Slack if you want. Thank you again, Gregor. We're over time, but really
Okay,
appreciated
thank you.
hearing your
Sorry for
perspective.
being a bit long.
Oh, no worries. All the questions.
Okay, and I learned a lot, so thanks for the feedback.
All right.
Thank you so much.
Take care.
Bye-bye.
Bye.
Ciao.